My name's Gordon James, the underlined one there. I work for Unilever in the personal care division, specifically for the deodorants category. I'm going to talk about, you can see the title of the presentation, Axillary Malodour Control, our bacterial transporters, the next target, so an obvious link to CBMNet there. And thank you to the CBMNet and Jen in particular for the invite to talk today. Apologies for not saying that before I even started. I'll talk about some work that myself and Diana have done at Coworth, although in reality the personal care group is based at our main lab in the UK lab in Port Sunlight, and also about a collaboration with Gavin Thomas's lab in York, and particularly this chap, Dan Bodden's excellent PhD work, which he's just completed in writing up, which really, and it's not an understatement to say so, has kind of changed, has caused almost a paradigm shift in our understanding of which microbes cause malodor and how they actually go about it. Okay, so why are we, Axillary malodor is a technical term for smelly armpits. So basically, as my children describe me, as an armpit scientist. So why do we need armpit scientists? We need armpit scientists because it's a huge business. So the global deodorants industry is valued at around 10 billion euros. And Unilever is, to get the old hat on here, it's not many sectors I can say this about, but we are overwhelmingly the market leader in deodorants with greater than 40% market share, and that is by organic growth. We do grow this sector by about 10%, double digits every year on year for the last over a decade now. And it's sales driven, and it's driven by our three main brands, Rexona, Axe and Dove. Rexona, you'll know, is Sure in the UK, Axe as Lynx in the UK, and Dove's obviously a master brand that's in Deo and various other segments as well. But the current product technology is based mainly on three things: aluminium containing antiperspirant salts, which are to reduce sweating, but also act as very effective antimicrobials at the levels we use them. And also antimicrobial solvents such as ethanol in aerosol sprays, in Axe Lynx sprays, and glycols in sticks. And also fragrance, and it's a really fragrance-driven industry. But so there is a demand for more targeted intervention, so that's really where we're coming at it here. What's it all about? What's the microbiology and the biochemistry about? Well, like all bodily odours, it starts with very innocent secretions that don't actually smell. This is the apparatus in the skin that causes it all. So this is a schematic of the underarm, skin surface here. This is the pilosebaceous follicle, the hair follicle. The eccrine gland secretes straight onto the skin surface. That's what gives us wetness. That's the classic sweat that we perceive as wet. And it's aluminium salts that reduce that. This is the sebaceous gland, goes into the hair follicle, and this is the key one for odour, is the apocrine gland, which also feeds into the hair follicle before coming up onto the skin surface, and is present only in the underarm and a few other locations, such as the areoli, the ears, bizarrely, and genitalia. But the key here is, even though these are the secretions here, although this one's the key to malodor, it's apocrine sweat is odourless. What causes the odour is the microbiota, the bacteria that live in our skin. So there's Staphylococci, there's Corynebacteria, there's Propionibacteria. There are others, that question mark shouldn't be there. And that produces, these precursors get converted to molecules that cause axillary odour, smelly armpits, underarm odour, whatever you want to call it. And traditionally it's been viewed that these are 16-androstenedione steroids, bizarrely, believe it or not, I'll come back to that shortly, volatile fatty acids, VFAs, and thioalcohols. What are the bacteria that cause it? Well, this is a study we did about a decade ago. Now what we tend to do is we use quite modern molecular techniques to do microbiology. We take a sample, we don't culture the bacteria, we did in this study. We don't culture the bacteria, we just smash them open, extract their DNA, amplify the 16S gene, and stick it into a sequencer, and then it gives us a profile of what's there. So that's a new technique that we use for microbiological analysis now. But this has gone back a few years when we did it by culturing. So you isolate, you take a sample from the armpit, and you basically culture it on a whole load of selective media, and you dilute them down, and then you count back to work out how many were there. You do it in a lot of people. You average the data out. You sniff their armpits. We have trained sniffers at Port Sunlight, believe it or not, because there is no other way of assessing how smelly an armpit is. There's no objective analytical tool that is as sensitive as a human nose And there are animals that are more sensitive, but they tend not to talk and give Malodour scores. So we use trained human assessors. So they tell us a Malodour score. We look at the numbers of each of the bacterial types in the underarm, and we look at which ones have got an association with Malodour. And you can see this is consistent with the literature, that the strongest associations with the Corynebacteria, or aerobic cryoniforms as they were called about a decade ago. So what are the molecules that cause underarm odor? That's the microbiology. As far as the biochemistry, I mean, there's a lot of literature on these steroids, 16-androstenedione, so-called because they've got this unusual double bond in this 16 position here. And there's a lot of hype around these at the time because they're actually pheromones in other mammalian systems, mainly pigs. So they were heavily implicated in axillary malodor, but analytical and biochemical studies now suggest otherwise. They have high levels of specific anosmia. A lot of people can't smell them at all. Few axillary bacteria actually can convert non-odorous steroids into odorous steroids. You can't find any evidence of association with axillary odor at physiological levels. These things are present, but they're at picomolar levels. And when you do analytic organoleptic studies, you show no link. Even when you see the 16-androstenes, you don't see a link with Malodour. So this is when you stick an injection down a GC column, it splits. Half of it goes to a mass spec to tell you what's there. The other half it goes to a human nose to tell you if it's an association with Malodour. And they never ever come through. So this is the biochemistry, but we no longer think this is relevant. This is relevant. Volatile fatty acids. The short and medium chain ones are among the causal molecules. There's various metabolic routes to them. We know that Corynebacteria are the subgroup called the A subgroup, partially degrade structurally unusual long chain fatty acids that are in sebum and apocrine sweat. We used to think, if you read my papers from about a decade ago, I'm absolutely adamant that's the main route to Malodour. It isn't. We've retracted it since in this later paper. We now think the main route to short chain volatile fatty acids is the conversion of amino acids, aliphatic branched amino acids such as leucine, to branched fatty acids such as isovaleric acid. But those aren't the main volatile fatty acids involved in underarm Malodour. They are there, but they particularly, things like isovaleric acid particularly drive smelly feet, foot Malodour. They've got a cheesy odour. There's these other volatile fatty acids, like slightly bigger and more complex in structure. 3-methyl-2-hexanoic and 3-hydroxy-3-methyl-hexanoic are the main ones. And they're actually in the apocrine secretion as acylglutamine precursors. And there's an enzyme in, well, certainly one Corynebacterium species that is a little bit up for grabs, this one. But there's an N-acylglutamine aminoacylase. It's basically an enzyme in some axillary bacteria that lyses these glutamine conjugates and releases the volatile fatty acids. And that is one of the two key routes to Malodour in most people's armpits. But as I said, the causal bacteria are not fully defined. And these are the other key malodorants. They're thioalcohols or mercaptal alcohols, whichever you want to call them. Basically, they've got a hydroxy group and a thiol group and an alkyl backbone. When we first discovered them, and there was various publications, and we were working on it as well, and there's several of them you can see. So there's 3-mercaptylpentanol. Some of them actually smell not too bad. Some smell quite nice in some people's opinion. The ones that are methyl branched, like 2-methyl-3-mercaptylbutanol and 3-methyl-3-mercaptylhexanol in particular, smell really nasty and do have a really characteristic smelly armpit note to them. Where do they come from? Well, if you look for biological precedents, there's a few about in wine. Typically cat urine, this is one, a molecule called felinine, produces a thioalcohol called, this thioalcohol, 3-mercaptyl-3-methylbutanol, very similar to some of the ones here. So we postulated, and others did as well, that the precursors were similar to felinine, and these are cysteine conjugates. And in fact, we did find one of the cysteine conjugates in sweat, as well as these various thioalcohols as well. However, things have moved on. There was an alternative hypothesis that was first proposed by one of the fragrance houses, Firmenich, and they said that the conjugates were actually cysteinoglycine conjugates. So they actually, the story here is that the biochemistry starts in the apocrine gland as glutathione. So these are glutathione conjugates. The glutamate is lysed by the humans in the apocrine gland, and then what comes out is a cysteinoglycine conjugate. And this is definitely true because you can actually find the S-cis-gly conjugate of this molecule, 3-methyl-3-mercaptylhexanol, and it's present in sweat. It's efficiently cleaved by a Staphylococcus species, Hemolyticus, and they proposed that lysis is mediated by a single CS-beta lyase enzyme rather than having to take the glycine off first. Entner and Nash, and this is Givaudan, showed that the corynebacteria could do it as well, but they said it was two enzymes, a dipeptidase and a cystathionine beta lyase. As I said, you can measure high levels of these in sweat, so really the evidence really does point now at cysteinoglycine conjugates. So that is our understanding of axillary malodor, and if I click on one further, those are the key pathways. So the main pathway for short-chain VFAs is the conversion of branched amino acids by staphylococci. But the two main pathways that come from apocrine precursors are the acyl-glutamine hydrolysis by possibly corynebacteria, possibly other bacteria, and the lysis of hydroxyalkyl cysteinoglycines by corynebacteria and staphylococci into thioalcohols. So in terms of moving the things on to more specific inhibitors and more specific intervention routes, that actually probably doesn't need to go through, that just says the same thing as I said. So let's move it. Instead of things moving it on and thinking about transport proteins, where do we go? Well, what we did is we considered this as a novel route. The idea here clearly is this has to get into the bacterium. So this is the cysteinoglycine conjugate of 3-methyl-3-mercaptylhexanol. There has to be a transporter, we think, or it has to get into the cell. Postulate it's a transporter. And then within the cell, there's either a single-step CS-beta lyase or a double-step with a dipeptidase removing the glycine first, then CS-beta and a CS-beta lyase to produce the 3-mercaptyl-3-hexanol. Then that has to actually get out the cell, either by diffusion or by active transport. So what we did is we focused Dan's PhD here to start with. And he did some screening work initially. This is a model substrate that you can buy off the shelf from Sigma. It's a cysteinoglycine conjugate of benzomercaptan, and it's a very nice model substrate, we thought, for measuring CS, the ability and capacity for a bacterium that we've isolated from the underarm to produce thioalcohols. And Dan did the screening, as we've done a lot of previously in Unilever, and he showed lots of corynebacteria were positive, a few staphylococci were positive. It tended to be more corynebacteria than staphylococci. So that fitted quite nicely with the hypothesis. But then Dan and Gavin said, really, if we're going to try and isolate transporters, we can't be using model substrates. We need to use real substrates, which of course you can't buy off the shelf. So we got them synthesized. And then we did this, Dan did this work instead. So now he's working with the cysteinoglycine conjugate of 3-methyl-3-mercaptylhexanol. So this is arguably the most physiologically relevant Malodour precursor, bar none. And when he did the screening, actually, and this is of typical axillary corynebacterium and staphylococcus isolates, similar to the ones that are very positive on the artificial substrate, and not an awful lot comes through. And this is I'll not jump back, this is a smaller scale, so partly driven by the fact that this thiol gives less of a reaction with the dye detection system, but it is definitely lower conversion rates. And we're not seeing an awful lot happening here, and we're not seeing an awful lot happening here. So what we did is we looked at not atypical, but some other corynebacteria and staphylococci that are found in the underarm, but not quite at the same levels as those ones we've been working on. And we did use actual isolates fresh from axillary washes. Same study, and you get more positive results with the corynebacteria. This is just 2 time points here. So these are creeping up. But what really came through was these staphylococci. So previously, these were all flatline, either negative or very, very low. But when you use the physiological substrate, all of a sudden you start seeing some of these axillary staphylococci producing really high levels of thioalcohol and doing it quite quickly. And this kind of, as I said earlier, it's a little bit of a step change in understanding. We didn't really expect this. In fact, my initial reaction was, to send them back in the lab because I thought he'd got the axillary, the Crinobacteria and Staphylococci mixed up. But it's been repeated several times, and Diane's done it as well. It's absolutely definitely the case. So is it active transport? Well, yes, it is. And you can see here, there's a range of Staphylococcus strains here, and here again, less so, but several. And what we've done here, what Dan's done here, is he's looked at a couple of inhibitors. So this is an uncoupling agent, CCCP, which always reminds me of my days as a student because it was the Soviet Union thing, but it's carbonyl cyanide metachlorophenohydrazone. I always remember that. This is sodium orthovanidate, which is an inhibitor, an ATPase inhibitor. You can see in both cases that both inhibitors wipe out the ability to produce bioalcohol, consistent with this being an active transport to take the precursor up. And Dan's moved it on massively since then. It's not my job here to stand up and give a presentation on his PhD, but in summary, he's cloned, expressed, and characterized the bioalcohol precursor transport protein Staphylococcus. It's present, it's really interesting, it appears to be present in all Staphylococci, certainly all where the genome has been sequenced, including non-thioalcohol producers. However, what's different is that in odor-forming Staphylococci, there's a putative CS lyase aminotransferase, which is unique to those species, which is also present. So we think what's happened is the transporter is a brilliant target, but it's not the rate-limiting step. The rate-limiting step is the actual whether the bacterium has the enzyme or not. So the plans are afoot to develop a transporter screening assay and to further characterize this putative CS lyase. So in terms of moving forward, obviously I'll put it to the floor to you guys. Where can we go next? So efflux transporters for thioalcohols and Staphylococci remain unidentified. If I go back to that slide, get there eventually, we don't know if that requires an active transport system. If it does, then that potentially is another target. And likewise, we don't know anything about the glutamine VFA conjugate transporter, and likewise we don't know anything about the VFA efflux system. I mean, this is volatile fatty acids charged molecules. Instinctively, to me, that means there must be an active transport system to get them out of the cell. So there's considerable scope there for targeting bacterial transporters as a Malodour control strategy. That's where I'm coming from here, because we've done some of this already. We're continuing down that route. We want to do more. There's several other targets we can go for. But if there's some other thoughts around the room, then I'm very receptive to them. I would also remind some of the discussions I've had earlier that one of my roles is a sort of science leader for antimicrobials. And okay, this is a clever approach, but most cosmetic biocides, antimicrobials, are basically membrane perturbing agents. They're cationics. They bind to set bacterial membrane surfaces, and they disrupt them, in inverted commas, in a way that no one really seems to quite fully understand. And when you see results that say, why is that cationic biocide better than that cationic biocide? Nobody really knows the answer. So a better understanding there, leading to the development of better antimicrobials, is another area that we would be interested around the realm of membranes. I think that is indeed it. So thank you very much.